one of the most important infectious diseases of family Equidae all over the world, equine viral arthritis, EVA, in 7 parts. So we can say that equine viral arthritis disease is a contagious subacute of family Equidae. Takes its name from the character of vascular pathology. There is no breed immunity from this disease. Equine viral arthritis disease characterized by the following. Number 1, acute respiratory signs. Number 2, abortion in pregnant mares. Number 3, necrotizing vasculitis of small arteries, which leads to edema in various body parts. Also, equine viral arthritis disease characterized by signs of skin rashes, infertility in infected stallions, and deaths of young foals. History of equine viral arthritis disease. The virus causing equine viral arthritis was firstly identified following an outbreak of respiratory disease and spontaneous abortion in a horse farm in Ohio, United States of America in 1953. First outbreak of equine viral arthritis reported in India was in 1989. The first outbreak of equine viral arthritis in United Kingdom was reported in 1993, and the outbreak affected 6 premises and around 100 horses were infected. Further spread of the virus was prevented by movement restriction. In United Kingdom, equine viral arthritis is a notifiable disease. Equine viral arthritis disease has afflicted equines worldwide for centuries. It has been more common in some breeds of horses than others in the United States. Epidemiology of equine viral arthritis. Pathogenic risk factor of equine viral arthritis. Pathogenic risk factor of equine viral arthritis is belonging to viruses, Rhelium riboviria, phylum Enteritidis, order Nidoviralis, superfamily Coronavirus, family Artriviridae, subfamily Ecortrivirini, genus Alpha Artrivirus, species Alpha Artrivirus equidae. Arteriviruses are spherical, small, and enveloped animal viruses with 50 to 65 nanomil particle with an icosahedral core that contains a single-stranded positive sense ribonucleic acid RNA molecule of about 12.7 kilobases. Icosahedron is a polyhedron with 20 faces. The name comes from ancient Greek meaning 20 seat. Arterivirus family includes, in addition to the alpha arterivirus equidae, porcine reproductive and respiratory syndrome virus PRRSV, lactate dehydrogenase elevating virus LDV that infect the mice, simian hemorrhagic fever virus SHFV which infect monkeys. This is a picture of alpha arterivirus equidae under electron microscope. This is a 3D structure of alpha arterivirus equidae. This picture describes the genome organization and virion structure of alpha arterivirus equidi, where the envelope appear here, and then E proteins, and then the ORF5, what's named as 5 leader and 9 reading frames, and then the 6 envelope protein GB2, GB3, GB4, GB5, M and N protein, and then the nucleocapsid protein N and the RNA of the virus. The equine viral arthritis virus genome includes a 5-leader sequence and 9 open reading frames, ORFS. The two most 5 proximal ORFS 1A and 1B occupy approximately 3/4 of the genome and encode 2 replicates protein PP1A and PP1AB. These two precursor proteins are extensively processed after translation into at least 12 non-structural proteins NSP1 to 12 by 3 viral proteases, non-structural protein 1, non-structural protein 2, and non-structural protein 4. The greatest variation in the equine viral arthritis virus replicase gene occur in the protein of ORF1A, encoding the non-structural protein 2 protein with considerable variation at amino acid 388 to 488. The structure proteins of the equine viral arthritis virion include 6 envelope proteins E, GP2B, GS, GP3, GP4, GP5, GL, and M, and a nucleocapsid protein N, which respectively are encoded by ORFS 2A, 2B, and 3 to 7 located at the 3 proximal quarter of the genome. These structure proteins are expressed from 6 subgenomic viral messenger RNAs that form a 3 coterminal nested set and contain a common leader sequence of 24 nucleotides encoded by the 5th end of the genome. 3 of the minor envelope proteins, GP2B, GP3, and GP4, form a heterotrimer in the equine arthritis virus particles, and the M and GP5 proteins form a disulfide linked heterodimer. The greatest sequence variation in the ORFS encoding structural equine viral arthritis virus proteins occur in the ORFS 3 and 5, which encode GP3 and GP5 respectively. GP5 expresses the major neutralization determinants of equine viral arthritis, and although considerable variation exists in the sequence of the GP5 protein of field strains of the virus, there is only one serotype of equine viral arthritis, and all strains evaluated thus far are neutralized by polyclonal antiserum raised against the virulent Buceras strain. Field strains of equine viral arthritis are frequently distinguished based on their neutralization phenotype with polyclonal antisera and monoclonal antibodies. Similarly, geographically and temporally distinct strains of equine viral arthritis differ in severity of the clinical disease they induce and in their apparent genetic potential. Although strains of equine viral arthritis from North America and Europe share as much as 85% nucleotide identity, these viruses generally segregate into clusters reflective of their geographic origin after phylogenetic analysis. Equine viral arthritis virus sensitivity. Equine viral arthritis virus inactivated by heat lipid solvents such as ether and chloroform. Equine viral arthritis virus sensitivity for different degrees of temperature appear as it remained viable for 75 days at 4 centigrade, 20 to 30 minutes at 56 centigrade, 2 to 3 days at 37 centigrade, at minus 20 centigrade for 5 years, and minus 70 centigrade for 60 years. Distribution of equine viral arthritis. Equine viral arthritis is a worldwide disease of equine that causes multi-respiratory disease and abortion. This is a map of world distribution of equine viral arthritis at May 2020, where there are different colors at the left of the map, as light green report the never reported countries with this disease, dark green disease absent, yellow is the disease suspected, red infection or infestation, the pink color is disease present, the disease limited to one region with purple color, where here United Kingdom, Germany, and Holland, as well as United States, Canada, are reported for the disease, while Morocco, Tunisia, and a lot of areas in Africa and Asia are never reported with the disease, and the disease is absent. Egypt, Sudan, Kenya, and some parts of Indonesia, Singapore, Brasilia, and Alaska are never reported. Russia have disease limited to one region. This is a map of world distribution of equine viral arthritis at May 2022. This is a map of world distribution of equine viral arthritis at May 2022. Animal risk factor for equine viral arthritis disease. Equine viral arthritis infect all the family Equidae, including horses, donkeys, and mules. Since the first diagnosed in 1953 in Ohio State, United States of America, all the family Equidae across the world are susceptible to the disease. Abortion rate of 50% in pregnant males exposed to the virus for the first time. It has been reported in all breeds of horses. All ages and sexes are susceptible. The disease will spread rapidly in susceptible horse population, resulting in high morbidity and low mortality rates. The stand-bred horse population found that 70 to 90% of pregnant mares who has been exposed to the virus will be aborted. Sources of infection. Sources of infection to equine viral arthritis disease will be all excretions and secretions of infected animals, semen of infected stallions for years, aborted materials such as fetus, placenta, and fluids during abortion or during birth. All of these act as a sources of infection to non-exposed or to new animals populations. Transmission of equine viral arthritis. Equine viral arthritis disease transmitted through inhalation of infected nasal droplets, venereal transmission by infected stallion, or by AI semen from infected stallion to susceptible mares, and transplacental transmission. This chart explains the transmission of equine viral arthritis in the breeding farms and how the equine viral arthritis outbreaks occur, where in breeding farms venereal transmission through the carrier stallion as natural reservoir of equine viral arthritis by 10 to 70%, through natural breeding, artificial breeding as shipment of contaminated frozen or shelled semen and frozen embryos, or horizontal transmission, lateral spreading through movement of horses or shipment of infective semen and embryos, through masturbation of the horses in the farm, or during semen collection through contaminated artificial vagina and phantom, personnel, teaser mare, nurse mare vomits, and respiratory air droplets. Equine viral arthritis outbreaks, no carrier status found between mares or gelding and foals less than 6 months. Horizontal transmission leads to equine viral arthritis outbreak through respiratory aerosols, viral insecretions and fetal tissues, personal equipments, and vomits. Vertical transmissions occur through congenital infection of late-term fetuses through movement of horses or shipment of infective semen and embryos from infected places or infected areas to non-infected places or non-infected areas or zones. Pathogenesis of equine viral arthritis disease. Incubation period of equine viral arthritis disease will be different according to the sources of infection and the way of transmission. Following respiratory spreading, incubation period will be 2 to 3 days, while post-venereal spreading is usually 6 to 8 days and may be up to 14 days in some cases post-venereal spreading. Post-respiratory infection, equine viral arthritis virus replicates in the pulmonary macrophages and then proliferates in macrophages and endothelial cells of the lung and the bronchial lymph nodes, from where it rapidly spreads in the circulation to endothelial cells and macrophages throughout the whole body. Equine viral arthritis viruses replicates mainly in the macrophages and epithelial cells. It causes necrosis of intestinal cells and muscle cells in the arterial media, causing advential edema, which leads to lesions occur in small arteries of approximately 0.5 micrometer in diameter, especially in intestine, spleen, lymph nodes, and adrenal capsule. Better understand of equine viral arthritis virus host interaction in the reproductive tract and the viral mechanisms of immune evasion at the site of persistent infection, characterization of the host mucosal antibody response to equine viral arthritis virus during persistence is of utmost importance. Hypotheses about pathogenesis of equine viral arthritis virus denote to their escape from antibody-mediated neutralization in the reproductive tract and consequently continues to be shed in semen of infected stallions. In this hypothesis, characterization of mucosal antibody isotype response to equine viral arthritis virus during persistent infection and identified presence of equine viral arthritis virus specific neutralizing immunoglobulins in the seminal plasma. The further demonstration that equine viral arthritis virus employs a complex mechanism of the immune evasion during persistent infection in the reproductive tract with successful viral escape from systemic humoral and mucosal antibody responses, as well as local inflammatory responses. Although endothelial cells and macrophages clearly are principal sites of equine viral arthritis virus replication, selected epithelial cells, mesothelium, and smooth muscle cells of the media of the arteries and the myometrium also may be infected. Systemic panvasculitis is a characteristic histologic feature of severe viral arthritis, although equine viral arthritis virus infection of pregnant mares can result in abortion, pathogenesis of fetal infection, and the mechanism responsible for abortion are not adequately defined until now. Developing to an immunohistochemical staining procedure utilizing equine viral arthritis specific monoclonal antibodies, and to use this procedure to localize equine viral arthritis virus in the tissues of an experimentally infected pregnant mare and her fetus may give an answer about the pathogenesis in the genital tracts. This is a schematic diagram of equine viral arthritis pathogenesis. Equine viral arthritis virus spread through inhalation of respiratory droplet, venereal transmission between mares and stallions, transplacental from the infected mares to the foals, where the virus, after multiplication, will cause viremia, then goes to blood vessels, causing severe vascular damage in small arteries, eyes, intestinal tract, visceral lymph nodes, adrenals, and ureters. This leads to hemorrhagic enteritis, blindness, pulmonary edema, and/or abortion. Death due to dehydration and anoxia is reportable. This schematic diagram shows clinical and laboratory findings and sequential pathogenesis of equine viral arthritis post-respiratory infection. Vertical bars correspond to the chronologic occurrence of respective clinical or laboratory findings and the distribution of the virus in body tissues and secretions. Fever here noted between the days of 2 days up to 12 days. Lymphopenia reported at the 4th day post-infection up to the 10th day. Edema reported at the 5th day up to 9th day. Neutralizing antibodies reported from the 6th day of infection up to 40 days. Nasal secretions noted from the 3rd day up to 14th day. The puffy coat and the virus present in them reported between the 1st day and 19th day. Presence of antibodies in the serum reported between the 4th day up to 7th day. The virus shed in the semen beginning from the 7th day up to 40 days. A portion noted between the 9th day of infection up to the 30 days post-infection. Natural and experimental equine viral arthritis infection induce a robust and protective immune response. The innate immune response has not been comprehensively characterized. Recent studies have demonstrated that equine viral arthritis virus inhibit type 1 interferon production and thus subverts the innate immune response. It has been shown that three non-structured proteins, non-structured protein 1, 2, and 3, have the capacity to block interferon synthesis with noticeable that non-structured protein 1 excreting the strongest effect. Post-infection, both equine viral arthritis, specific complement-fusing (CF), and neutralizing viral antibodies develop with production of long-lasting immunity and protection from reinfection with most viral strains. Both complement-fusing and viral neutralizing antibodies developed between 7 and 14 days post-infection, then peak either after 2 to 3 weeks or 2 to 4 months post-infection respectively. Complement-fusing antibodies steadily decline by the 8 months post-infection, while viral neutralizing antibodies persist for 3 years or more. Both structured proteins, GB5 and M, and the non-structured protein 12 induce humoral immunity. Structured protein 5 contains 4 major neutralization sites A, B, C, and D. Structured proteins 5 and M heterodimerization is essential for induction of antibody responses and protection. Despite presence of high neutralizing antibodies, the equine viral arthritis virus persists in the reproductive tract of carrier stallions. Viral evolutionary mechanisms take place during persistent infection in the reproductive tract, leading to the emergence of genetic variants with distinct neutralization phenotypes due to amino acid substitutions in the major viral neutralization sites, and thus conferring the ability of the virus to successfully escape humoral immunity. Foals born to immune mares are protected against clinical equine viral arthritis by passive transfer of viral neutralizing antibodies in the colostrum. Viral neutralizing antibodies appear in the serum of newborn foals a few hours after colostrum intake, level peak at age of first week, and titers decline to extinction at the age of 2 to 6 or rarely 7 months. The mean half-life of equine viral arthritis maternal antibodies in serum from foals is about 32 days. The last experimental study at 2018 about the cell-mediated immune response to equine viral arthritis virus infection cleared that it induces a specific cytotoxic CD8 T lymphocyte, and their precursor may last for at least a year following infection. Clinical severity of equine viral arthritis infection of horses varies greatly. in equine population. How to spot equine viral arthritis disease? One or more of the following clinical signs spotting presence of the disease: abortion, conjunctivitis, swelling of testicles or other, swelling around eyes and lower legs, fever and runny nose, anorexia, lethargy and stiff movement. Rare cases show severe clinical signs and deaths in young foals, while many infected horses show no clinical signs. Clinical severity of equine viral arthritis infection varies greatly. The vast majority of infections are inapparent, especially those that occur in mares bred to persistently infected stallions. Outbreaks of clinical disease caused by equine viral arthritis viral infection are characterized by one or more of the followings: abortion of pregnant mares, fulminant infection of neonates leading to severe interstitial pneumonia or enteritis, and systemic illness in adult horses with any combination of leukopenia and pyrexia, respiratory signs with nasal and ocular discharges, peripheral edema, hives, and persistent infection of clinical signs observed in natural cases of equine viral arthritis vary considerably among individual horses and between outbreaks, and depend on factors such as the age, physical condition of the horses, its challenge dose, route of infection, strain of the virus, and environmental conditions. Although there is only one serotype of equine viral arthritis, the clinical disease produced by different virus strains ranges from severe lethal infection caused by the horse adaptive B. Cyrus strain to clinically inapparent infection. Very young, old, debilitated, and immune suppressed horses are predisposed to severe equine viral arthritis disease. Regardless of the infecting viral strain, the vast majority of naturally infected horses recover uneventually from equine viral arthritis disease. Young foals, however, can develop fatal infections that lead to severe fulminating interstitial pneumonia, and foals up to few months of age can develop a rapidly progressive pneumoenteritis syndrome. With the notable exceptions of abortion and fulminant respiratory disease in foals, mortality rarely, if ever, occurs in natural outbreaks of equine viral arthritis. The highly virulent horse-adapted Bacillus strain of equine viral arthritis, which causes high mortality in healthy adult horses, is not representative of field strains of the virus and is best regarded as an aberration. A picture of depressed horses suffering from equine viral arthritis during feverish conditions. A depressed horse suffering from equine viral arthritis during fever. A depressed and recumbent horse suffering from equine viral arthritis. An infected horse with equine viral arthritis during fever, showing recumbency and depression. Recumbent mare and foal suffering from equine viral arthritis during fever. A horse suffering from coughing during respiratory manifestation due to equine viral arthritis infection. conjunctivitis in a horse diseased with equine viral arthritis, conjunctivitis with excess lacrimation, blepharospasm in a horse diseased with equine viral arthritis, conjunctivitis and edema, pink eye in a horse diseased with equine viral arthritis, conjunctivitis and purulent vitreous with keratitis and opacity of aqueous humorous in a horse diseased with equine viral arthritis, conjunctivitis with excess lacrimation in a horse diseased with equine viral arthritis, conjunctivitis and edema, pink eye in a horse diseased with equine viral conjunctivitis and edema, pink eye in a horse diseased with equine viral petechial hemorrhage at upper gym of a donkey diseased with equine viral arthritis, urticarial type of skin rash because of blood vessels lesion in a donkey infected with equine viral arthritis, urticarial type of skin reaction because of blood vessels lesions in a neck and the front thorax in a horse diseased with equine viral arthritis, edema in the ventral abdominal wall may involve the prepuce and the scrotum in a diseased horse with equine viral arthritis, edema in the ventral abdominal wall may involve the prepuce and the scrotum in a diseased horse with equine viral arthritis, edema in the ventral abdominal wall may involve the prepuce and the scrotum in a diseased horse with equine viral arthritis, edema in the ventral abdominal wall may involve the prepuce and the scrotum in a diseased horse with equine viral arthritis, limbus edema in a donkey infected with equine viral arthritis, gastrointestinal tract disturbance due to equine viral arthritis virus infection, showing diarrhea, abdominal pain, jaundice may be reported. Abortion reported 12 to 30 days post-exposure to infection and occurs during the 3rd to 10th month of gestation period. Abortion reported alone, without, or with general illness. No reports about placental retention. Abortion without placental retention. Aborted feti without placental retention. Abortion without general illness and placental retention. Abortion without placental retention. Abortion without placental retention. Prognosis of equine viral arthritis. Uneventual recovery in majority of adult horses can cause abortion and mortality in neonatal foals, as well as carrier stallions shed virus in the semen. Post-mortem lesions in equines dead due to equine viral arthritis disease are the following: huge edema of the eyelids, edema with petechiation in lung tissues, hemorrhagic enterocolitis, splenic infarction, laboratory diagnosis of equine viral arthritis. Samples collection. Samples have to be initiated as early as possible post onset of fever and or other clinical symptoms. This table showing sampling, testing, and handling by means that type of samples and the tests done for them, how to ship them to the laboratory with handling of these samples. Number 1, nasopharyngeal washing or swabs. This used for RT-PCR or viral isolation. Shipped with leak-proof container and the handling is chilled overnight. Number 2, EDTA or saturated whole blood. No heparin used. This for RT-PCR or viral isolation tests. Shipped in leak-proof container, as well as handling with chilled overnight. The third sample is the semen. This used for RT-PCR or viral isolation. Shipping in leak-proof container with chilling or frozen overnight. The fourth sample is fetal membranes or fetal tissues such as lungs, liver, kidneys, and spleen. This used for RT-PCR or viral isolation or fluorescent antibody technique. Shipping in leak-proof container, chilled or frozen overnight for handling. The fifth samples is sera, double-paired serum, acute and convalescent stage. This used for serum neutralization test. Shipping with red top tube or proof container. Handling by chilled overnight. Viral isolation and fluorescent antibody technique. Immunoperoxidase test and serological examination by complement fixation test, agar gel immunodiffusion test, ELISA, and viral neutralization test. RT-PCR, where the positive band appear at 395 base pair. Sequencing of the equine viral arthritis virus for detection of the phylogenetic tree, where red lines here denote to the equine viral arthritis virus and the year of its isolation. Histopathological examination, where intranuclear inclusion bodies and the nuclear pyknosis are apparent. Vesiculitis is a characteristic lesions in cecum, colon, spleen, and associated lymph glands. Immunohistochemical identification of immunoglobulin positive epithelial cells in the accessory sex glands of stallions persistently infected with equine viral arthritis. A and B, where IgA specific epithelial immune staining in ampulla in A and vesicular gland in B. C, IgG1 specific epithelial immune staining in the vesicular gland. In D, IgG4 on 7 specific epithelial immune staining in the vesicular gland. Differential diagnosis of equine viral arthritis. Equine viral arthritis have to be differentiated from equine influenza, equine herpes virus 1 and 4, equine rhinitis virus A and B infections, purpura hemorrhagica, equine infectious anemia, allergic reactions and toxicosis from ingesting Hori aleosum and equine viral rhinopneumonitis, African horse sickness, and leptospirosis. Prevention and control of equine viral arthritis. Preventing of equine viral arthritis through application of good biosecurity, testing stallions, teasers, and mares before breeding time, application of vaccination program, isolation and testing of newly purchased horses, isolation and testing of any horse who when suspect in any clinical sign. Detection of infected equines by using of fluorescent antibody technique where seropositive animals prevented from mating and seronegative animal mating only. Avoid introduction of infected equines to free areas or barns. Vaccination by using of attenuated modified live virus vaccine which had been used in United States and Canada since 1985. It is safe and efficient. This vaccine administered intramuscular, inducing protective immunity in vaccinated animals. This vaccine elicits viral neutralization antibodies within 5 to 8 days post-vaccination, with a peak between 7 to 14 days that may last for 2 years. Reactivation boosts the serological response, providing protective immunity for an extended time. Annual reactivation with this vaccine are recommended as established in equine viral arthritis control and prevention guidelines. Maybe febrile reaction had been found in some vaccinated population, about 50%, with transient lymphopenia. Stallions required live attenuated virus vaccine, while mares require killed whole virus vaccine. Seropositive stallions restricted from teasing, mating, or AI unless RT-PCR tested. A picture of equine arthritis vaccine. Persistent cheddar should be castrated and serology should be performed on imported horses. Mares are tested within 4 weeks of mating. Clinical disease of equine viral arthritis is notifiable in United Kingdom. In an outbreak, isolate and restrict movement until antibody negative for 1 month. Release animals from isolation post 4 weeks from the last case of equine viral arthritis was confirmed or if viral detection tests are negative. The whole barns and surrounded areas with their facility should be thoroughly disinfected.